Hi friends, good afternoon. How are you? Hope all are fine. Welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Today we shall discuss about the life cycle of Chlamydomonas. Chlamydomonas is a motile unicellular green algae. It reproduces by asexual method and sexual method. Asexual reproduction occurs by the formation of different types of asexual spore like zoospore, aplanospore, hypnospore and pamela-stage. The sexual reproduction can be isogamous, anisogamous or primitively oogamous depending upon the species. So first of all, let us discuss about the process of asexual reproduction in Chlamydomonas. In Chlamydomonas, asexual reproduction occurs by the formation of zoospore, aplanospore, hypnospore and pamela-stage. First is zoospore formation. Asexual reproduction by zoospore formation is the common method of asexual reproduction in Chlamydomonas. Zoospore formation occurs when the condition is favorable. At the time of zoospore formation, the flagella are withdrawn, so the cell become non-motile, the contractile vacuole disappear, and the protoplast gets separated from the cell wall. This protoplast undergo longitudinal division, thereby forming two daughter protoplasts. These two daughter protoplasts undergo successive longitudinal division at right angle to each other. This results in the formation of 4, 8, 16 or more number of uninucleate daughter protoplasts. These uninucleate daughter protoplasts develop a wall around them. They also develop flagella and also contractile vacuole. So these uninucleate daughter protoplasts become motile. These daughter protoplasts are known as zoospore. Zoospore are similar in shape and structure to the parent Chlamydomonas. The difference is that zoospore are small as compared to the parent Chlamydomonas. The zoospore are released from the parent Chlamydomonas cell either due to gelatinization of the cell wall or due to rupture of the cell wall. The zoospore, they can grow and form a new Chlamydomonas. Second is aplanospore formation. Asexual reproduction by aplanospores occur when the condition is not favorable. For example, in case of drought. At the time of aplanospore formation, flagella are withdrawn, so the cell become non-motile and the protoplast divide repeatedly to form 2 to 16 daughter protoplasts. These 2 to 16 daughter protoplasts, they don't transform into zoospore. They remain inside the parent Chlamydomonas. All these uninucleate daughter protoplasts, they secrete a thin wall around them. This thin walled daughter protoplast transform into aplanospore. In some species, for example in Chlamydomonas nivalis, when there is severe drought, the aplanospore develop a thick wall around them. These thick walled structures are called as hypnospore. Aplanospore as well as hypnospore can undergo germination when the condition is favorable to form a new cell. Last one is pamela stage. Pamela stage is seen in case of adverse condition. So, in this pamela stage, the protoplast present inside the parent Chlamydomonas divide to form 4 to 8 daughter protoplasts. This daughter protoplast can also divide again to form a large number of daughter protoplasts. All of them remain enclosed in a common mucilaginous matrix. This temporary colony of hundreds or thousands of cells that are enclosed in a common gelatinous matrix is known as Pamela stage. In some species, for example, in case of Chlamydomonas nivalis, all the cells of the Pamela stage develop thick wall around them. These thick wall cells are called as hypnospore. The wall of the hypnospore contains a pigment known as hematochrome. The pigment hematochrome imparts red color to the hypnospore. And when these red colored hypnospores are present in large number, they are responsible for the phenomenon known as red snow. This Pamela stage is a non-motile stage and it is a temporary reproductive phase in the life cycle of Chlamydomonas. When the condition becomes favorable, all the daughter protoplasts that remain enclosed in the matrix, this daughter protoplast transform into motile zoospore, and the zoospore can grow further to form a new Chlamydomonas. This is about the process of asexual reproduction in Chlamydomonas. Next we shall discuss about sexual reproduction. Sexual reproduction in Chlamydomonas takes place in the deficiency or absence of nitrogenous compound in the medium. It can be isogamous, anisogamous or primitively oogamous depending upon the species. So first of all, let us discuss about isogamy. Most of the species of Chlamydomonas are isogamous. In this process, the male gamete and female gamete are morphologically similar. They are uninucleate biflagellate structures. They are usually naked, that means they don't have cell wall. Such type of morphologically similar gametes are called as isogametes. These isogametes are formed due to the repeated division of the protoplast of the parent cell to form 8, 16, 32 or 64 daughter protoplasts which undergo transformation into isogametes. Some of the isogamous species are homothallic, whereas remaining isogamous species are heterothallic. In homothallic species, fusion occurs between gametes which are formed in the same parent cell, whereas in heterothallic species, fusion occurs between gametes that are formed in the parent cell of two opposite strains. Fusion occurs between two isogametes at their anterior end, and this fusion leads to the formation of a diploid zygote, and the zygote undergo meiosis that results in the formation of 4 haploid zoospore which can mature to form a new Chlamydomonas. This is about isogamy. Next is anisogamy. Anisogamy is a process in which the male gamete and female gamete are morphologically dissimilar. The male gametes are comparatively smaller and they are called as microgametes, and the female gametes, which are comparatively larger, are known as macrogametes. The macrogametes are formed due to the division of the protoplast of the parent cell that leads to the formation of 2 or 4 macrogametes. The macrogametes are also called as female gametes, whereas the microgametes are formed due to the repeated division of the protoplast of the parent cell that leads to the formation of 8 to 16 microgametes. So the microgametes are formed due to higher number of division of the protoplast of the parent cell. The male gametes are called as microgametes and the female gametes are called as macrogamete. As in isogamy, in anisogamy also fusion occurs between one microgamete and another macrogamete, at their anterior end. In Chlamydomonas, few species are oogamous. In this process also, the male gametes and the female gamete are morphologically dissimilar. The male gamete is known as microgamete and the female gamete is known as macrogamete, which is equivalent to egg or ovum. At the time of formation of microgamete, the protoplast of the male cell divide repeatedly to form 16 daughter protoplasts, and the 16 daughter protoplasts transform into 16 uninucleate biflagellate microgametes. The microgametes are smaller and they are more active. Whereas at the time of formation of female gamete, the female cell withdraws its flagella and directly function as a female gamete, which is non-motile. Fusion occurs between male gamete and female gamete. Fusion occurs at the anterior end that result in the formation of a zygote. Next is germination of zygote. After fusion between male and female gamete, the wall at the point of fusion dissolves, and this quadriflagellate zygote swim for some time and then settle down. It secretes a primary wall around it. At this stage, the zygote has two nuclei, two chloroplasts, and two eye spots. In next 24 hours, the two nuclei fuse that result in the formation of a diploid zygote. Before germination of zygote, the diploid nucleus divide meiotically to form 4 haploid nuclei. These 4 haploid nuclei, along with a bit of cytoplasm, transform into 4 haploid zoospores. In heterothallic species, out of 4 zoospores, 2 zoospores are of plus strain and remaining 2 zoospores are of minus strain. These zoospores come out of the zygote and the zoospore can develop to a new Chlamydomonas. This is about sexual reproduction in Chlamydomonas. In the life cycle of Chlamydomonas, the zygote is the only diploid structure. Remaining all are haploid. So the life cycle of Chlamydomonas is an example of haplontic type of life cycle. Thank you friends. Please share, like and subscribe this video. Thank you very much.